So we have the data in the correct format for rROMA. What we also need is the gene sets. So the gene sets, they can be very different things. They can be pathways. They can be groups of genes that have the same function. They can be groups of genes that are regulated by the same transcription factor. They can be very diverse. So there are websites where you can find gene sets. You have to take a look at the tutorial to get more information. One example of such a website is the Path Guide website. So it contains a whole list of resources that contain pathway information or molecular interaction information. Not all the links are working and not all the links are very recent, but it is a very extensive list, as you can see here. And when you click here on one of the resources, like this one, for instance, you are redirected to the website of that resource. And on many of these websites, you can download pathways or gene sets. And as you can see, you can download the gene sets from here in what is called GMT format. And this is the format that rROMA accepts. GMT format is just a kind of file. It's a top delimited text file, basically with a specific format. You can find more information here. The link is provided in the tutorial. So it's just a text file that contains a number of columns, and the columns have to be in a specific order and they should contain a specific type of information. So you can see here what it looks like. So every row represents one gene set. And you see here that indeed in most of these resources, the gene sets are represented by gene symbols. And this is why we had to convert our ensemble IDs into gene symbols. So if you are going to work with this and you're going to create or download the GMT files yourself, then rROMA has a function that allows you to import them. And I'm not going to actually use this function, but I can show you here the function, this one, read GMT file. So this allows you to import a GMT file into R so you can use it for rROMA analysis. Now there is another resource of gene sets that is very well known, and that is this one. The molecular signatures database that was created by the Broad Institute. So for human and mouse, they compiled several collections of gene sets that can be used for functional analysis. So you have the human collections here and you have the mouse collections here. Many different collections. So you see that genes are grouped according to their position in the human genome. They come from pathway databases or publications. You also have here regulatory gene sets, so linked to microRNAs or targets from transcription factors, for instance. You have cancer-related gene sets. You have another one here, the oncogenic signature gene sets. So here you have groups of genes that show the same expression behavior when you're going to look in samples that are related to cancer, so tumor samples and corresponding controls, obviously. You have the ontology gene sets, so linked to gene ontology. You have immunologic gene sets. And here you have cell type specific gene sets, so gene sets that are expressed or specifically expressed in specific cell types. So many different types of gene sets are available. We are going to work with these, the hallmark gene sets. They're kind of an overview, a curated compilation of all these. So you have hallmark gene sets for human, you also have hallmark gene sets for mouse. Basically, they are the same, the hallmark gene sets. Yeah, they contain exactly the same genes. The only difference is that the gene symbols here are written in capitals because it's human, and here the gene symbols are written in small letters because they are mouse, but they contain the same gene sets with the same genes. If you want to, you can download the gene sets here. You see here, download GMT files, perfectly possible. I'm going to take these, for instance. So I click here. You can see here the different gene sets. You can even click a gene set. And then you can download it here in GMT format. I will also briefly show you the hallmark data sets here, the gene sets for here. Also here the same thing. You can click them and then you can download them in GMT format. So if I wanted to, I could download them from here. However, there is an easier way in rROMA. So rROMA has a function that allows you to access the gene sets from this molecular signatures database. So that's what I'm going to do. So I'm going to select gene sets from this database and I'm going to ask him to retrieve all the gene sets that have the word hallmark in their name. So which gene sets will I retrieve in that case? Well, as you can see, I will retrieve the hallmark gene sets. So that's what we do here. So 50 gene sets with variable numbers of genes, as you can see here. And you see that their names always start with the word hallmark underscore. I'm going to remove this hallmark underscore, because it will only make the name of the gene set larger and it will not look nice on figures. So I am going to remove that, and that's what I do here. Yeah, so I'm going to remove this part from the names of the gene sets.